Good evening, everyone. My name is Meredith Chewy, and I'm the Conferences and Events Manager at the American Nutrition Association, the ANA. The American Nutrition Association is very pleased to be hosting Dr. Jack Gilbert tonight to discuss the human microbiome in health and disease. Dr. Jack Gilbert is a professor in the Department of Pediatrics at UC San Diego and the Scripps Institution of Oceanography. His past research includes a focus on ecology, evolution, and metabolic dynamics of microbial ecosystems from environments including built environments, oceans, rivers, soils, air, plants, animals, and humans. He is the co-founder of both the Earth Microbiome Project and the American Gut Project, as well as the co-author of Dirt is Good, The Advantage of Germs for Your Children's Developing Immune System. Welcome, Dr. Gilbert.
It's my pleasure to be here. My name is Jack. I'm a professor of pediatrics and oceanography at University of California, San Diego. Previously, I was the director of the Microbiome Center at the University of Chicago and Argonne National Lab in the Marine Biological Laboratory. And I've just moved over my lab in the last 3 or 4 weeks, so I'm getting used to these new titles. I'm going to talk to you today about the human microbiome in health and disease. So the human microbiome consists of the microbial communities that are resident in our bodies. They've evolved alongside us. There are some 38 trillion microbial cells resident in our body and on our skin and in our mouth and in our lungs. Compared to our 30 trillion human cells, they just about outnumber us. They contain a huge amount of genetic information, and they produce an inordinate amount of metabolites, which can affect how our body processes food, all the way through to how our immune system functions and responds to a changing environment. The microbiome is made of up to 1,000 different species spread out over the body. They inhabit virtually every mucosal surface, which is open to the outside world. We believe, although it's hard to prove, that there could be around 10,000 different strains of microbes, especially of bacteria, present inside each human. And I say there are 38 to 40 trillion individual cells per person. The way we analyze the microbiome is to determine their association to disease are what we call microbiome-wide association studies. These are akin to genome-wide association studies that leverage obviously human genes and try and associate them through epidemiological investigations to individual disease states or response to drugs. Microbiome-wide associations look for statistical relationships between either bacterial organisms, their genes, their functional gene pathways, or the metabolites they produce to determine very discrete associations between those and diseases, between those and responses to drugs, between those and the onset or exacerbation of disease. In fact, the key elements by which microbes can actually affect disease, it comes through their metabolism. Microbes are little chemical factories. They produce an inordinate amount of, an inordinate diversity of chemical products, such as here on the right you can see acetate, which is a short-chain fatty acid. Acetate is the predominant short-chain fatty acid produced by bacteria after the fermentation of long-chain carbons, so long-chain fibers. And acetate is also incredibly important in moderating satiety. So it changes how hungry you are. An increase in the amount of acetate in your bloodstream can actually influence, after it passes the blood-brain barrier, influence satiety. So we think that could be important in mediating dietary calorific intake. They can also produce other amino acids such as tryptophan, lysine, and we found that through investigations in a cohort of population study based out of the University of the West Indies with Terence Forrester in Jamaica, that bacteria producing an excess amount of amino acids can actually play a role in how people respond to dietary protein restriction. So when people don't get enough protein, sometimes they can have a deficit of protein in their body, which can lead to them catabolizing their muscle mass to rebalance the equilibrium between the amount of protein coming in and the protein requirements for the body. we've seen that in certain situations, that catabolism can fuel microbial synthesis of amino acids, which actually rebalances that equation. But only in those individuals who have access to microbes which can respond to the catabolic breakdown products like urea and have the ability to synthesize excess amino acids and make them available to the host. However, this could be a very discrete role in how microbes mediate dietary responses. We know that microbes can also catabolize certain types of drug. Some drugs called prodrugs are actually developed to be metabolized or altered by microbes so that they actually have a host beneficial effect. But other drugs, such as acetaminophen, can be broken down by certain bacteria to produce things like acetaminophen sulfate, which can actually be damaging to the liver. So, we need to understand that. Bacteria can also break down things like bile acids, primary bile acids represented by the Ca here, and DCA-based deconjugated bile acids, such as secondary bile acids. So bacteria mediating that relationship between primary to secondary bile acid metabolism can be extremely important in protecting the host from pathogens, things like Clostridium difficile infection. If we see spores of C. diff in the gut, a buildup of primary bile acids can actually lead to the germination of those C. diff spores. And obviously, if you have enough bacteria in your gut that are breaking down those primary bile acids to secondary bile acids, those C. diff spores maintain spore form and do not germinate. We think this could be incredibly important and a reason why things like antibiotic therapy may be responsible for the onset of certain C. diff infections. One of the key strategies here, and what my good friend John Alverdi gave to me, is a unidentifiable quote from Jorge Luis Borges, who stipulates that everything touches everything. In Luis Borges' literature, alterations in one of his characters has huge rippling effects throughout his novels that touches all the other characters in his literary universe. And similarly, in the body, an alteration in the microbial metabolism inside your gut, for example, could have huge concomitant effects upon very distal effects zones inside the body, having uncharacteristic or unconsidered relationships to human health. In this stunning study from Rothschild et al., published last year, they demonstrated that certain human characteristics, such as HDL cholesterol, glycemic status, response to food index, can actually be predicted more effectively if we include the microbiome, represented here in green, in our assessment of a human population. So just understanding a person's age, their gender, and their calorific intake, we can predict to a certain degree of success their HDL cholesterol, their waist circumference, their hip circumference. We're pretty good. But if we add microbiome data into there, the ability of their bacterial community to moderate certain metabolic responses, we get a significant improvement in our ability to predict some of these host-based characteristics. Adding genetics in there doesn't necessarily improve upon the microbiome. Therefore, we think that microbiome may actually be a more important role in some of these host metabolic disease states than host genetics. And in fact, we can only predict with about 50% accuracy if somebody is going to be obese or not based on their genetics. But we can predict with almost 95 to 100% accuracy if someone's going to be obese based on their microbiome, suggesting the microbiome plays a significant role in mediating that metabolic disease. Where do we get our microbiome from? Predominantly, we obtain our microbiome from our mothers. It's one of our first and earliest birthday presents. In this study from Maria Gloria Domingos Bello back in 2011, she demonstrated that children born by vaginal birth and C-section actually acquire a different microbial colonization event or an early microbial consortia within the first few days of life. What you're seeing here is a principal coordinate plot. Every dot on there, every circle, is a microbial community. And the closer those dots are together, the more similar those microbial communities are. So at the top, you can see all the microbial communities associated with the oral cavity of different mothers in this cohort. On the left, you see in red mother's vaginal microbiota, and on the right, you see in blue mother's skin microbiota. And then you see babies' gastrointestinal microbiota born by vaginal delivery or C-section delivery, clustering or being more similar to mother's skin or mother's vaginal cavity, respectively. We think this is a potential early alteration or early effect for immune activation in the child, which will be differentiated based on how that child is born. So your earliest birthday present can vary depending on whether you're born through the birth canal or whether you are born by a C-section delivery. This differentiation leads to us all being quite microbially unique as far as we are aware. At least from the 700,000 microbiome samples that have been processed in individual people around the world, we do not appear to have identified a microbial doppelganger for anybody in those cohorts, suggesting that there is a high degree of variability within individuals. This is valuable for obviously precision or personalized medicine, because if the microbiome plays a role in drug response, efficacy, or dose-dependent activation, or in disease onset or exacerbation, then understanding the microbiome could be incredibly important for being able to provide that precision or more precise personalized treatment strategy. Even identical twins tend to have a unique microbiome, although statistically they are more similar to each other than non-identical or normal siblings. We do believe that the microbial world is being eroded. So here I'm obviously showing the Amazon rainforest. If we remove the trees from the Amazon rainforest, we will see a significant impact on local hydrology and on global Amazon basin hydrography, and potentially therefore on local and global climate. So the removal of species from a system has an impact upon the ecology and far-reaching unintended implications upon much broader categories of activation. Same is true for the human gut. We tend to term a disruption of the microbiome something called dysbiosis. It's a much maligned term. So here I'm taking a figure from Bindlos et al., published in Mucosal Immunology last year, which I think is highly effective in demonstrating a definition for dysbiosis compared to a state of homeostasis, whereby the microbial community in the gut is in equilibrium with the body. When it is in equilibrium, the microbiome is predominantly anaerobic. It is fermenting some of the fibers that the human is consuming through their diet, and they're producing short-chain fatty acids among many other compounds, short-chain fatty acids like butyrate. Butyrate is highly effective in fueling beta oxidation and respiration inside the colonocytes, allowing colonocytes to mature. And as they do that, they actually consume oxygen out of the bloodstream, and preventing that oxygen from bleeding through into the gastrointestinal tract. So they maintain this positive feedback mechanism, allowing for continuation of the anaerobic status of the gut, and therefore continuation of the anaerobic organisms which are mediating the production of the chemicals which are fueling that cycle. Dysbiosis, however, is usually predicated on the removal of some of those obligate anaerobic organisms, and a proliferation of the facultatively anaerobic organisms, which can handle oxygen potential. And what we tend to see there is then a reduction in the production or the fermentation of fibers, and therefore the production of short-chain fatty acids like butyrate, and a reduction in the maturation of the colonocytes, which increases the transition of oxygen out of the blood vessel across the gut barrier and into the gastrointestinal tract, raising oxygen potential, and thereby inhibiting the redevelopment or distribution of those obligate anaerobes, leading to a negative feedback loop whereby some of these facultative anaerobic bacteria become super dominant And many of them produce things like lipopolysaccharide, LPS, and other MAMPs and PAMPs, molecules which are recognized by the human body as pathogenic or inflammatory, causing an increase in local inflammation and then systemic inflammation, and potentially leading to the onset of certain diseases. That gives you a definition for dysbiosis and homeostasis, which, while gut-centric, at least provides a clear strategy for microbial interaction with the host, and something we can really understand and measure. The microbiome has been explored in many different disease states. I believe there are over 600 ongoing clinical trials, either completed and being analyzed or still recruiting. If you look at clinicaltrials.gov and type in microbiome, you can find that information. And they cross many different disease states, everything from cancer and IBD all the way through to diabetes and cardiovascular disease. And therefore, there's maybe an opportunity for people to go, well, look, microbiomes has been looked at in everything. And therefore, we want to be careful about trying to oversell it as being important. But I do believe it is important in many different disease states. I think that disease states which were otherwise not linked or for which the links were not understood or identified, disruption in microbial metabolic activity and its host microbe interaction could be extremely important in helping us to identify those missing links between disease states, looking towards maybe closing the gap in our understanding of how different diseases are connected. One of the very original microbiome therapeutic strategies involved the use of a fecal microbiome transplant. In this study from 2011, a small group of individuals who were suffering from recurrent Clostridium difficile colitis were put in a randomized trial group where they were either given vancomycin or vancomycin with a bowel lavage as the standard mechanism of treatment for recurrent C. diff, which got a 23 to 30% success rate. Or but they were given an infusion of donor feces, a so-called fecal microbiome or fecal matter transplant, whereby bacterial populations in the fecal matter of a healthy individual were transplanted into the colon and large bowel of an individual that was suffering from recurrent C. difficile infection, leading to over an 80 to 90% success rate. And this is not a new therapy. An ancient Chinese textbook provides us with evidence that this was being used as far back as the 6th century in a term called yellow soup or dragon broth, whereby the fecal matter from a healthy person, especially a healthy child, was given to people with recurrent diarrhea as a mechanism to rebalance the qi in the gastrointestinal tract. Ergo the microbiome. We think this microbial re-establishment or repopulation of the microbial community inside the gut may be incredibly important in helping to alleviate diseases that have a dysbiotic origin. Although there are many disease states where fecal microbiome transplants have been tested, such as IBD, where it has not proven to be effective in the treatment of that disease. We also, in my previous role at the University of Chicago, I was a professor of surgery, although I have no MD and they don't allow me to use knives. We were very interested in the role that bacteria could be playing in surgical complications, especially surgical infections. And the understanding that dysbiosis of the gastrointestinal tract prior to surgery could be highly effective in predicting somebody's likelihood of developing a surgical infection took hold as a potential strategy for helping people to improve their outcomes post-surgical post-surgery. So one good indication of this, as we talked about earlier, is the idea of the gastrointestinal tract being predisposed to a pro-inflammatory system, which is then exacerbated by the way we treat the gut and the body during the prep for surgery. So if you have a baseline microbiota which is affected by your lifestyle, how much you get outside, how much exercise you do, as well as what kind of pets you have in your home, as well as your diet, most importantly, represented here by a hamburger and fries. The microbiome in your gut is probably going to be highly affected by that. So if you eat a very poor diet, for example, rich in saturated fats and sugars, you're likely to actually promote the growth of some of those facultative anaerobic organisms that are pro-inflammatory. This could lead to a significant reduction in the mucosal membrane, the mucous barrier, and an increase in the probability that some of those bacteria could exacerbate injury post-surgery. And then, of course, we pump the gut full of antibiotics. We give the patient IV antibiotics to try and clean it out as much as possible. Many of those pro-inflammatory facultative anaerobes are actually resistant or resilient to some of those antibiotics and therefore aren't cleaned out by that process. And then when we actually perform the surgery, we almost select for those superbugs which are more likely to be able to attack the gut and potentially negatively affect recovery. In an ideal situation, your microbial functional potential would potentially be affected by surgical practice, but recover quickly after surgical practice. And that's ideally what we're looking for in our patients. And in this study, we demonstrated in the animal model, this is work with John Alberdi and Olga Zabarina at the University of Chicago. We demonstrated that a surgical practice can actually select for some dangerous organisms. So in this case, rats were given a 30% hepatectomy, and they were injected with an intrathecal injection with a strain of Pseudomonas aeruginosa. 24 hours later, we selectively grew Pseudomonas aeruginosa on plates, and we got these two separate types of colonies, smooth colonies and wrinkled colonies. Interestingly, those smooth colonies, when injected into sham animals, had no impact at all upon the survival rate of the animals. But the wrinkled colonies, which were much more abundant post-surgical practice, actually had a very high mortality rate, killing 100% of the animals within about 17 hours. This suggests that the actions of surgical practice in the animal and the stress that surgical practice causes may also be causing stress to the bacteria and therefore could be exacerbating surgical injury and surgical infection during practice. To illustrate that, we can look at the number of stresses which surgery practice performs. So let's say we're doing a gastrointestinal resection. We're pumping the gastrointestinal tract then full of oxygen as we cut into it, and that obviously doesn't bode well for those obligate anaerobes. And we pump the body full of antibiotics, and that selects for only those organisms which can survive. And then we stitch the gut up, and the body then starts to suck nutrients from the gut in order to help repair itself as a stress response, starving certain types of bacteria which panic, activate nutrient acquisition strategies, which can then lead to the production of a biofilm which grows on the wound site. It releases collagenases, breaking down that gastrointestinal wall as the collagen degrades and causing the gastrointestinal contents to leak out into the body cavity, which can therefore lead to septic shock and sepsis and death. Therefore, we believe that there are multiple stresses that the microbial community feels. If you have a poor diet prior to surgery, you're going to probably have selected already for organisms which will respond in this kind of way to surgical practice. Therefore, from a dietitian's perspective, enabling people to make healthy dietary choices prior to surgical practice may be immensely important in reducing the likelihood that they will suffer a surgical infection or post-surgical complications such as ileus and many other states during surgery. What do I think has been happening in the rest of the world? Well, we used to play outside where we were exposed to lots of microbes. Now we play inside where we're not exposed to very many microbes at all. We tend to use cleaning products to eradicate as many organisms as possible, both potentially good and potentially bad bacteria. We think this has reduced the exposure of microbes that our immune system is seeing, and that may actually be leading to our immune systems becoming oversensitized to the microbial world. Potentially by increasing our microbial exposure, we might actually be able to stimulate immune responses which protect our bodies from those kind of immune responses which could lead to disease. In a study we performed and published in New England Journal of Medicine with Carol Ober and Anne Sperling and Donato Facelli back in 2016, we explored the potential role of microbial exposure in farmyard settings in the protection of children against the development of asthma. As has been shown in many studies from Germany, the microbial exposure that people get on farms can actually play a significant role in predicting asthma response rates or asthma onset rates. And so we wanted to determine in a controlled group of individuals in the United States whether this same phenomena was true. We chose the Amish and the Hutterites, which are both European groups of individuals which migrated in the late 1700s and mid 1800s to North America. The Amish set up shop between Pennsylvania, Ohio, and Indiana, and various other places, and the Hutterites now live up in the Dakotas. The Amish tend to have individual personal family farms where the front door is 50 yards from the barn door and the children work on the farm, whereas the Hutterites tend to live in household communities and then have an off-site farm which only the working men attend to, severing the relationship or the exposure that children would normally get to that farming environment. Other than that, they have the same genetic predisposition towards the development of asthma. They tend to eat very similar, tend to be organic diets, lots of fermented foods, etc. And they tend to have a reduction in their dependence upon technology. But the Amish have around 4% asthma in their population, and the Hutterites can have up to 25% asthma in the population, almost 2.5 times the US average. So this tends to suggest a dichotomy, an epidemiological dichotomy between these populations. If we look at the immune system of those children on these farms, the Amish tend to have more neutrophils and less eosinophils. The Hutterites tend to have less neutrophils and more eosinophils. Interestingly, the Hutterites tend to have older, more sticky neutrophils, and we can see that in the CXCR4 and CD11B ratios here, which are cell surface markers on those neutrophils. This would tend to suggest that the Amish children are being exposed to more antigenic substrates in the environment because they need more neutrophils in circulation compared to the Hutterites. But the Hutterites, while they have neutrophils being expressed, they tend to be older, suggesting those neutrophils are staying in circulation, not seeing and therefore reacting to foreign objects very often, which makes them produce these cell surface markers like CXCR4. They become stickier, they tend to bind together, and therefore they have a more pro-inflammatory response when they finally see a relatively benign antigen. This, we believe, leads to an increased inflammatory potential, which in a predisposed individual could lead to the onset of asthma. To test this hypothesis, we took anaerobic mice with Donato Facelli from Arizona. Sorry, germ-free mice. If they're anaerobic, they would die. Germ-free mice, and exposed those mice to dust from either Amish children's bedrooms or dust from Hutterite children's bedrooms. And then we looked at their eosinophil production rate, their neutrophil production rate, when exposed to evalbumin, a protein they have been sensitized to. And you can see in yellow there the eosinophil rate upon exposure to just evalbumin is very high. But however, if we expose these mice to dust from Amish children's bedrooms, their eosinophil rate drops very significantly down to essentially no allergic reaction levels. Whereas the exposure to Hutterite dust shows no protective effect whatsoever. The neutrophils, when exposed to Amish dust, are much, much higher. And this tends to mirror, if I go back one slide, what we see in the children's immune systems. The Amish have higher neutrophils and lower eosinophils, much the same as we see in mice exposed to the dust these children are breathing in while they sleep. So we think this is indicative of a potentially important immunological response to external environmental microbial exposure. We, from work from a colleague in Colorado, Reber et al., published in PNAS in 2016, there's also interesting evidence to suggest that certain types of soil bacteria that could be exposed to from people in the environment, like Microbial vaccae, could actually reduce anxiety-like behaviors and promote more aggressive behaviors in animal models. So, for example, in the left graph here, we see that animals in the presence of a subordinate mouse, when exposed to Microbial vaccae in red here, will show a significantly increased response to spending time out in the open, suggesting that they are more less anxious and more willing to explore the world. And mice that are just exposed to a subordinate mouse tend to show a significant increase in proactive behaviors, suggesting a decreased anxiety upon exposure to a subordinate animal. So, potentially exposure to bacterial antigens from soil environments could be playing a very significant role in how animals respond, even emotionally, to the world. Microbes can also play a significant role in the onset of obesity. In one study from Liping Zhao's group, published in ISME journal in 2012, a moderately obese man weighing 385 pounds lost 113 pounds by just embracing a diet of traditional Chinese whole grains over a period of about 9 to 12 months. He looked at his microbiome before and afterwards and found afterwards there was one bacteria which was particularly abundant at the beginning, which had dropped below the level of detection after this after this diet window. So he changed the microbiome in his gut, and certain organisms were no longer competitive under this new diet. When that bacterium was grown up, Enterobacter cloaceae B29 it had a significant role in increasing the obesity coefficient for mice. So mice exposed to Enterobacter cloaceae B29 in their gastrointestinal tract actually increased the amount of fat that they developed with the same diet compared to an animal that had no Enterobacter cloaceae B29 in it. And significant work from the likes of Ruth Ley, who's now works at an institution in Germany, and groups down such as Jeff Gordon in the University of Washington University in St. Louis in the United States, have demonstrated there are many different types of bacteria which can both protect against obesity, protect against an obesogenic diet, for example, and ones that can also exacerbate obesity on an obesogenic diet. So if you respond to an obesogenic diet, such as a high-fat, high-sugar diet, by increasing the amount of fat deposition you generate, then it's highly probable that you have certain types of bacteria which are actually impacting that. And in fact, we've demonstrated through the work of Vanessa Leone and Eugene Chang in collaboration with those two at University of Chicago, that the microbial community in the gut tends to respond to a high-fat diet, represented here by HF, by promoting the growth of bacteria, especially proteobacteria, some of these facultative anaerobic organisms which don't ferment fibers, but which do produce a large amount of hydrogen sulfide. And that hydrogen sulfide tends to bleed across the gastrointestinal barrier into the bloodstream and can actually affect circadian rhythm disruption in the liver, which can lead to an increase in obesity. We know that disruption of circadian rhythm is mediated by working nights or by getting jet lag, can lead from brain-derived alteration in metabolic potential, therefore increased obesity in people. But this also tends to suggest that the same mechanism of circadian rhythm disruption in internal organs affected by working nights and by having jet lag is also the same mechanism that that occurs when you eat a high-fat diet and you promote the growth of certain types of bacteria in the intestine. So I am on the scientific advisory board for a company called Day 2, which has commercialized research from Eran Elanav and Eran Segal's labs at the Weizmann Institute in Israel. And they developed a strategic opportunity to be able to predict glycemic response to diet based upon the microbiome. And so this is a personalized nutrition angle. I have no immediate financial relationship with Day 2. However, I am on the scientific advisory board, and there is a possibility of a financial relationship in the future. However, to talk about this, obviously, the human genome is very important, and people considered it to be vital in helping us to identify strategic opportunities to intervene in disease. But then the microbiome became a key component in 2010 after the release of the first results of the Human Microbiome Project sponsored by the National Institutes of Health. And now we're in a position to actually combine both components, your genome and your microbiome, to provide actionable precision medicine. That was an 8-year program. And the company Day 2 was founded in 2015 off the back of the research from the Weizmann Institute to actually be able to take these data and provide precision medicine advice. How do they do it? Well, they take measured personalized features, including the microbiome, including hemoglobin A1C blood tests, questionnaires on your lifestyle and what you eat, anthropogenic measurements, and a food diary. And they put all this via a base calculator developed by Eran Segal, a machine-learned algorithm that can be used to predict what kind of diet you should eat based upon your personal glycemic response to individual types of food. Remember, a glycemic response to a food, a type of food, is basically an average. So low glycemic foods that's an average low glycemic response, and your personalized response might actually be much higher to a low glycemic response than somebody else's. Obviously, an average has a distribution around a mean, right? And some of those distributions can actually be quite large. And so what this algorithm does, it utilizes the microbiome and to try and identify how your body is going to respond to those types of foods. And in doing this, they can actually explore how certain types of food may influence your glycemic response, and therefore give you an ability to be able to control your personalized glycemic response by avoiding foods to which you personally have a high glycemic response to. And this could mean that being able to guess the good diet just based upon our understanding of high or low glycemic response foods may not be possible. And you need to be able to identify that, eggs and bread, noodles and ice cream for you are actually fine. You might not be able to consume coffee and chocolate, marzipan and sushi. It might not actually be a good diet for you. And one of, I think this is Eran Elanav's results, showed that he could actually reduce the variability, the fluctuation in his glycemic response by going on a precision diet predicted for him, which significantly reduced the variability day to day, minute to minute, hour to hour in his glycemic reaction in his blood. Obviously, for a pre-diabetic or even a type 2 diabetic, this kind of information could be extremely important in helping to moderate the severity of the disease state. We think this is incredibly important, indicative of the fact that time matters a lot in our measurement of microbial communities. The green line here on the left graph represents your microbial diversity, or the disorder or the distribution of variability in your microbiome throughout your life. And when you're born, it's incredibly high. It can change day to day, hour to hour, as the microbiome community starts to settle down into a rhythm, which often happens between the ages of about 6 months and 24 months. But then it tends to maintain a very high level of stability throughout your life, represented by the green line. But day to day, that stability can actually be quite variable. So you're always you, but you show differences in the relative proportions of your membership day to day. And then hour to hour, we can see even bigger variability. Within a 24-hour cycle, for example, we can see a rather large amount of variability between morning and nighttime as the microbiome responds to certain types of food intake. We think this is the reason breakfast is such an important meal, because it actually allows the bacterial community to re-establish metabolic activity as you feed them the kinds of food inputs they require. And on the right here, we're actually seeing the microbial distribution over time of different organisms inside an individual person's intestine, showing huge amounts of variability. Just to riff on one more disease type, cancer, we know that environmental exposure and diet can play a very significant role in mediating the epidemiological rate of cancers. Here where I'm showing graphs representing Japanese living in Osaka compared to Japanese who have emigrated to Hawaii, and comparing those to Hawaiian Caucasians, suggesting that the American lifestyle, at least for Japanese people migrating to Hawaii, increased the rates of prostate cancer, colon cancer, and breast cancer, interestingly reducing the rates of stomach cancer, potentially due to the use of antibiotics to treat Helicobacter pylori infections, which are one of the predeterminate organisms associated with cancer onset. When we look at colon cancer, for example, we can identify bacteria present in the intestine of individuals who have colon cancer. The tree on the left is a microbial family tree. The organism, Fusobacterium nucleatum, highlighted in red, is significantly enriched in the fecal matter from people who have CRCs, colorectal cancer, and the ones in blue are significantly elevated in healthy cohort controls. And you can actually see in the histological image on the right here, highlighted in red, Fusobacterium cells that are uniquely co-associated with the CRC tumor tissue, suggesting a very tight relationship which modulates beta-catenin signaling and alters the immune visibility or the visibility of the tumor to the immune system, potentially activating tumorigenesis as well. And indeed, some probiotic therapies have been demonstrated in animal models to be highly effective in helping to mediate the efficacy of immunotherapies for treating certain types of cancer. So in this case, we're taking an animal model and injecting it with a metastatic melanoma cell line. And we can see a significant increase in tumor volume on the left here over 25 days of observation in untreated animals. If we give them alpha PDL1 in the dotted open circle line as an immunotherapy, imalzabub, we can actually see a significant decrease in tumor volume over the same time frame. And we see the same decrease in tumor volume if we just give the animal a bifidobacterial cocktail as a probiotic. And this is obviously potentially interesting because it suggests that the probiotic is stimulating immune responses and causing a decrease in tumor volume. And in fact, when the bifidobacterial probiotic BIF is combined with alpha PDL1 immunotherapy, we see a doubling down on the effect size for tumor volume decrease. This does not mean you should treat your cancer with probiotics. This is an animal model, and obviously is highly susceptible to the probiotic therapy. But there is research ongoing in several commercial entities to try and understand if this can be leveraged as a strategy for treating certain types of cancer. The microbial world around us is highly diverse, and you alone, sitting here watching this video, are emitting around 38 million bacterial cells and around 7 million viral particles into your immediate vicinity every hour, just sitting still. We live in a microbial universe, and our homes, our offices, our hospitals are being colonized continuously by the bacteria that we release. In fact, around 99% of those bacteria will probably die after they leave the warm, moist environs of your body and end up in our dry, cold building structure. But their DNA hangs around for a while. And certain organisms can actually survive reasonably well and maintain viability, i.e. they can be grown, after they have been deposited into these environments. Therefore, we're working with architects and designers to understand how to leverage that information and build healthier buildings where the microbial exposure people get inside those buildings is extremely important in mediating human health consequences. In fact, in 2014, we published a study where we demonstrated the colossal distribution potential of microbes inside buildings. Every single one of these edges in this Bayesian network represents the movement of bacteria over time between the noses, feet, hands, and surfaces in a home for individuals in a small family of 4 people. And the distribution was constant. Bacteria were constantly being moved between these surfaces, being deposited, picked up, redistributed, and shifted. In fact, when we added dogs into a home, we actually see a significant increase in the movement of microbes between people in a building, suggesting that dogs act as a super conduit for transferring microbes between individuals. And we know that couples that have a dog share more bacteria than couples that do not have a dog. And we also know that certain dog bacteria from work by Fujimura et al., published in PNAS in 2014, from the lab of Susan Lynch in the University of California, San Francisco, certain bacteria associated with dog stool, like Lactobacillus, can be used to desensitize mice to the development of asthma when taken as an oral administered organism. This is interesting. It suggests that certain dog bacteria may be playing a role in asthma prevention. And indeed, children that grow up interacting with a dog within their first year of life have a 13% reduction in the likelihood of developing asthma. We can even use microbes deposited by people as a forensic tool, and we're working with the National Institute of Justice to determine if this could be effective for criminal investigations, but we're not there yet. But we do know from this study, for example, if we look at the kitchen counter, the x-axis is time, and the y-axis is the percentage of bacteria found on that kitchen counter, or bacterial signals, that originated from one of 3 human occupants and 3 dogs. Person 1 is a 25-year-old man living with his mother and father. Person 2 in orange is his father, and person 3 in yellow is his mother. And you can see that for the kitchen counter, at least, might be a very gender imbalanced home, as the mother's microbiota is sometimes 100% of the bacteria found in the kitchen counter. And we can see the floors are predominantly dominated by bacteria from the dogs, and we do find bacteria from the dogs on the front doorknob and the kitchen light switch, probably not because they're touching them, but because there is transfer from your hands touching the dogs to those surfaces. And my colleague Jared Hampton-Marcel, down here on the bottom right-hand side with the fetching mask and hairnet, is actively working with criminal investigation units across the country to try and identify whether signatures left behind by burglars could be used to identify people who had broken into a space. And I'll stop there. We have myself and my colleague Rob Knight, also at the University of California, San Diego Department of Pediatrics, have published a public book called Dirt is Good, which is a question and answer book, almost like a quick reference guide to all the questions you could ever possibly have, starting in pre-pregnancy, pregnancy, and then during childhood, regarding the microbiome and health. And I encourage you to investigate that as an option for future reading. Thank you very much for your time.
Thank you, Dr. Gilbert, for that truly fascinating presentation. This has been an enlightening evening, and we invite our audience to learn more about your work at gilbertlab.com. Good night and best of health to you all.